Welcome everyone. So this is our last session today of this symposium about genetic admixture at Collège de France. And this last symposium is about ancient DNA studies and archaic integration and admixture. And so we have the pleasure to hear the first speaker, Irving-Pease, who will talk about methods for admixture-aware inference of natural selection in the Holocene West Eurasia. Thanks, Evan. The floor is yours.
Well, thank you, and good morning, everyone. Sorry if my voice sounds a bit scratchy. I'm a little bit under the weather at the moment. So my name's Evan, and I'm a postdoc in the Racimo group at the Center for Geogenetics in Copenhagen. And today I'm going to be talking about some methods we've been developing collaboratively with other groups to understand the interplay between natural selection and admixture over the last 10,000 years or so. But before I get into the details of our study, I'd like to kind of briefly motivate the work that we've performed. And that we've been looking quite closely at the Mesolithic-Neolithic transition because it was one of the most significant changes in human society with really far-reaching consequences for the diet, health and social organization of affected populations. And these dramatic changes in lifestyle were likely concomitant with shifts in the selective pressures exerted on a wide range of traits and newly emergent diseases. And whilst genome-wide association studies of present-day human populations have identified large numbers of genetic variants associated with phenotypes of interest, the extent to which these variants have been under positive selection during recent human evolution remains unclear. And here we've reconstructed the allele frequency trajectories and selection coefficients of tens of thousands of GWAS variants through time using a large panel of ancient genomes. And this work was performed in collaboration with the Mesoneo Consortium, led by S.K. Willerslev, Morten Allentoft, and Martin Sikora, who have compiled a panel of more than 1,600 ancient shotgun genomes, including 317 new Mesolithic and Neolithic individuals sampled from across West Eurasia through the Holocene that are part of this study. And you can read more about this data set and some of the analyses we performed in a recently released preprint on bioRxiv called Population Genomics of Stone Age Eurasia. So in work led by Barbara de Motor and Olivier Delanoux at the University of Lausanne, we phased and imputed our ancient shotgun genomes using the recently published software Glimpse. And this produced a final data set containing almost 4 million SNPs passing all quality filters from around 1,500 imputed diploid ancient genomes, which gave us the power to infer selection at really kind of unprecedented resolution and to disentangle the effects of admixture on those selection signals. So this quite kind of busy and complicated figure here comes from the GWAS catalog, and it shows genome-wide trait associations for each chromosome in the human genome grouped and colored by the class of trait. And so we took the entire GWAS catalog, containing more than 200,000 associations for in excess of 2,000 traits, and we used that to ascertain a list of genomic variants which contribute to a broad range of phenotypes, including disease risk in present-day populations. And we filtered this list to retain SNPs below a genome-wide significance threshold of 5 × 10⁻⁸, and a condition on the ancestral allele being known and the GWAS risk allele being present in our data set. And in total, we retained around 37,000 unique SNPs with these trait associations. And to investigate the extent to which GWAS SNPs are enriched for signatures of selection, we also ascertained frequency-paired control SNPs that were at least 50 kilobases from the nearest GWAS SNP or gene region. And one of the advantages of working with phased whole genomes is that we can use a class of methods known as ancestral recombination graphs to perform inference of selection. And here we use the recently published method Relate, developed by Leo Spiedel and Simon Myers from the University of Oxford. And in brief, we began by using Relate to infer an approximation of the ARG for all 1,000 genomes, for all samples in the 1,000 genomes phase 3 dataset. We then extracted subtrees for European populations of interest and used the iterative algorithm and relate to jointly infer a population size history and to re-estimate branch lengths. We then used a modified version of clues, a method developed by Aaron Stern and Rasmus Nielsen from the University of California, Berkeley, to infer allele frequency trajectories and selection coefficients using the ARG using the ARG inferred from present-day 1,000 genomes data, as well as a separate analysis using a time series of imputed genotype probabilities from our ancient samples. And this figure shows a Manhattan plot summarizing the results of the 1,000 genomes analysis. In the top panel are the negative log 10 transform p-values from the selection analysis of the GWAS variants. And in the bottom panel are the results from the frequency paired control SNPs. And what we observe here is that when using present-day data alone, we were unable to identify any genome-wide significant selective sweeps in either the GWAS or control sets. However, the GWAS variants were significantly enriched for evidence of selection with respect to the controls. On the other hand, when we ran the selection analysis using the time series of imputed genotypes, we identified 11 significant selective sweeps, replicating many previously reported signals of selection at well-known loci, such as those causal for changes in lactase persistence and skin pigmentation. And in the control group, we did not observe any selective sweeps, further confirming that GWAS variants are substantially enriched for evidence of selection. However, our ability to detect selection is influenced by population demography and episodes of admixture between populations. And one of the most interesting findings from the last 10 years or so of ancient DNA research is that present-day West Eurasians are a complex mixture of multiple different ancestral populations. This map here is showing the spread of farming, material culture, and ancestry into Europe. So to account for population structure in our samples, we applied a novel chromosome painting technique developed by Alice Pearson and Richard Durbin at the University of Cambridge. And this new method, using a technique similar to genealogical nearest neighbors, allows us to accurately assign ancestral population labels to haplotypes found in both ancient and present-day individuals. And this schematic shows a simplified demographic history of West Eurasia, in which present-day individuals can be modeled as a combination of 4 highly differentiated ancestral populations. That's Western hunter-gatherers, or WHG, Eastern hunter-gatherers, or EHG, Caucasus hunter-gatherers, or CHG, and Anatolian farmers, or ANA. And whilst these populations no longer exist in the present, the majority of genomic loci in present-day individuals can be traced back to an ancestry path which transited through one of these 4 populations. So by conditioning our selection analysis on one of these 4 marginal ancestries, we can use clues to infer selection trajectories in a manner that is approximately invariant to changes in the admixture proportions through time. This plot shows a composite of results from running clues on each of the 4 marginal ancestries. And here we identified 21 genome-wide significant selection peaks, highlighted in gray and labeled with the most significant gene within each locus. And within each sweep, SNPs are positioned on the y-axis and colored by their most significant marginal ancestry. Outside of the sweeps, SNPs show p-values from the pan-ancestry analysis and are colored gray. And strikingly, when the selection analysis is conditioned on each of the 4 marginal ancestries, we identified almost twice as many genome-wide significant sweeps compared to the pan-ancestry analysis. And we often find variants with strong allele frequency changes in one ancestral population, but not another, and find that analysing all ancient individuals without accounting for their ancestry composition leads to a decrease in our ability to identify selected variants and a blurring of the temporal signal of allele frequency changes. These results indicate that admixture between ancestral populations has masked evidence of selection at many loci across the genome. And to demonstrate this further, I'll zoom in on some of these sweep loci to show what is happening in closer detail. So this plot shows a zoomed in view of the MCM6 locus, the derived allele of which is responsible for lactase persistence in present-day Europeans. The top row in this figure shows Manhattan plots for the pan-ancestry analysis on the left, labeled all, and the 4 marginal ancestries on the right. And below each of the Manhattan plots are the allele trajectories for each of the ancestries, showing the most significant SNPs in this locus. And what's interesting is that the strong signal of selection we see in the pan-ancestry analysis is being driven largely by a sweep occurring in the Eastern hunter-gatherer background. However, the causal SNP for lactase persistence is not the most significant SNP in this ancestry. And we also observe that the lactase persistent SNP is not selected for in the Western hunter-gatherer background, but that another SNP associated with blood protein levels is. And this suggests that this locus may have experienced selection on more than one causal variant. And this is a pattern that we observe across many of the genome-wide sweep loci. Another locus where we found a strong signal of selection is at the FADS2 gene associated with fatty acid metabolism. And here we find that the strong signal of selection in the pan-ancestry analysis is driven primarily by a sweep occurring on the Eastern hunter-gatherer and Anatolian farmer haplotypic backgrounds. And interestingly, we do not find evidence for selection of this locus in the Western hunter-gatherer background. And most of the frequency rise in the Eastern hunter-gatherer background occurs after their admixture with Caucasus hunter-gatherers, where the selected alleles were already at close to present-day frequencies. And this shows that selection was acting at this locus at distinctly different time periods in each of the ancestral backgrounds as farming moved northwards and westwards across Europe. We also detected strong selection at the SLC45A2 locus, which is associated with skin pigmentation. And we find that the most strongly selected allele began rising in frequency around 13,000 years ago. And that selection at this locus shows a consistent pattern of increase across all marginal ancestries, differing somewhat in the timing of their frequency rises. That the Anatolian farmer ancestry background shows the earliest evidence for selection at this locus, followed by Eastern hunter-gatherers and Western hunter-gatherers, beginning around 10,000 years ago, and Caucasus hunter-gatherers 2,000 years later than that. In all ancestry backgrounds except Western hunter-gatherers, the selected haplotypes reached near fixation by around 3,000 years ago, whilst the Western hunter-gatherer haplotype background contains the majority of the ancestral alleles still segregating in present-day Europeans. We also detected a large 33 megabase wide selective sweep in chromosome 6, spanning the HLA region, which is highly enriched for genes associated with immune function. The selection trajectories of the variants within this locus support multiple independent sweeps occurring at different times and with differing intensities. And this highlights the complex temporal dynamics of selection at the HLA locus, which not only plays a role in the regulation of the immune system, but also has association with many other non-immune related phenotypes. And the high pleiotropy of this region makes it difficult to determine which selection pressures may have driven these increases in frequencies. However, profound shifts in lifestyle during the Holocene, including a change in diet and closer contact with domestic animals, combined with high mobility and increased population sizes, may well be responsible for strong selection pressures on loci involved in immune response. So due to time constraints, I've only been able to show you 4 of the 21 sweep loci that we identified, but I'd like to summarize some of our main findings. Firstly, that ancient DNA considerably improves power to detect variants under selection. Secondly, that admixture between ancestral populations has masked evidence for selection at many loci. And thirdly, that multiple selected alleles may be common within genome-wide significant sweep loci. And as I mentioned before, you can read more about this work in our preprint on bioRxiv. But what about complex traits? And if we return to our figure from the GWAS catalogue, showing the distribution of trait associations across the genome, we can clearly see that most traits of interest are highly polygenic, and with many containing hundreds of independent loci, each of which contribute to a small percentage of the trait's genetic variance. So one of the questions we were interested in asking was what complex traits were under selection during the last 10,000 years? And to investigate this, we've been using the polygenic adaptation likelihood method, known as PALM, to aggregate across the hundreds of independent SNPs associated with each complex trait, where each marginal SNP trajectory is weighted by its effect size for that trait. And I'm going to show you an example of this analysis applied to the autoimmune disease multiple sclerosis, for which we have more than 200 fine-mapped risk loci, many of which fall within the HLA region, where we previously saw such a complex pattern of ancestry stratified selection. So this plot visualizes the results of that PALM analysis, in which the x-axis represents time in units of thousands of years BP, and the y-axis shows the population level polygenic risk score rescaled to have a range between 0 and 1, such that a scaled PRS of 1 would mean the entire population was fixed for all positive risk alleles. And if you look closely, you'll see that this plot is made up of stacked horizontal lines, each of which represents the change in population frequency of the positive risk allele weighted by its contribution to the overall risk score. So each individual SNP trajectory is further colored by the significance of the p-value from a marginal selection test in clues, and then the stacked lines are sorted by their p-values and the direction of their effect on the trait, such that SNPs with significant negative selection are placed on the bottom of the plot, and SNPs with significant positive selection are shown on the top. And what we observe here is that of the 165 fine-mapped SNPs that are callable in our ancient imputed samples, only 21 of these SNPs have a marginal p-value below the Bonferroni corrected significance threshold, highlighted here in yellow. However, when we consider all 165 SNPs in a joint test using PALM, we observe a highly significant p-value of 1.87 times 10 to the minus 9, consistent with strong positive selection in favor of increased risk of multiple sclerosis. And the majority of this increase occurs between 6,000 years ago and 2,000 years ago. And it's important to caveat here that we're not arguing that multiple sclerosis is itself an adaptive phenotype, but rather that it seems likely that environmental pathogens associated with dietary and lifestyle changes during the Holocene led to positive selection for a more active immune system, which subsequently became maladaptive once those environmental pathogens were removed through the invention of pasteurization and reduced exposure to domestic animals in present-day peoples. So like the ancestry stratified trajectories I showed earlier, we can also perform this PALM analysis conditional on one of our 4 ancestry paintings. And here we see that the majority of the signal in the pan-ancestry analysis is driven by a steady increase in the scaled polygenic risk score for multiple sclerosis in the Caucasus hunter-gatherer background. Caucasus hunter-gatherers are one of the 2 main ancestral populations that contributed ancestry to the subsequent Yamnaya steppe pastoralist population. These results are consistent with selection favoring a strong immune system in populations with close proximity to domestic animals and consumption of large quantities of unpasteurized dairy products. Excuse me. We also observe evidence for positive directional selection in Anatolian farmers. However, the signal here is considerably weaker than Caucasus hunter-gatherers, despite there being more marginally significant SNPs in this ancestry background. And notably, we observe no evidence for positive selection in Western hunter-gatherers, who would have had substantially lower daily contact with zoonotic pathogens. In the case of Eastern hunter-gatherers, we observe that this ancestral background has the largest number of marginally significant SNPs in any of the ancestry paintings. However, the effects of these SNPs are counterbalanced by each other at the level of the population risk score. And we do not find evidence for directional selection operating on the trait within this ancestry background. And I think this plot nicely indicates how the strength of polygenic adaptation can be uncorrelated with the strength of marginal selection in the SNPs that contribute to that trait. So another way that we can look at this data is to visualize the contribution each SNP makes to the overall change in population level PRS, which is shown here in this rain cloud plot. So each dot represents the change in frequency of the risk allele at a particular SNP, weighted by its scaled effect size. And what we observe here is that there are many SNPs in all of our ancestry paths, which make a substantial contribution to changes in the population PRS over time. However, these SNPs do not achieve significance in a marginal selection test because their changes in frequency were too gradual to reject neutrality. So it's only by looking at the aggregate signal across all contributing SNPs that we can identify that directional selection is occurring within some of these marginal ancestries. Now, if we return to the Caucasus hunter-gatherer plot that we looked at earlier, we might consider the question of, well, why do we see such a strong signal of ancestry stratified selection, which persists for thousands of years after all four ancestral populations have become fully commingled? And we think there are two likely explanations for this. Firstly, that Caucasus hunter-gatherer ancestry continue to correlate with steppe pastoralist dietary and lifestyle practices, which increased exposure to environmental pathogens, maintaining the selective advantage of an aggressive immune system. And secondly, that selection favored the specific composition of risk alleles present in Caucasus hunter-gatherer haplotypes due to epistatic effects. And that many of these risk alleles are located in the HLA region where epistasis is well documented. So in conclusion, we can reconstruct the trajectories of population level polygenic risk scores while accounting for changes in ancestry. And we find that there's no correlation between the number of marginally significant SNPs and the significance of the polygenic adaptation test itself. We also find specifically that multiple sclerosis risk has been positively selected for, and that Caucasus hunter-gatherer ancestry is a strong predictor of multiple sclerosis risk. And with that, I'd like to thank all the funding bodies that have made this research possible and all the talented scientists who have contributed analysis and support, especially members of the Racimo group and the Center for Geogenetics. And I'd like to thank you, the audience, for listening.